All right, so we got some pretty kick-ass news about this, and I don't have to. I looked at the objectives again, and I don't have to teach you about recycling of hemoglobin, which is going to make our lives a lot easier. So what we're going to do now is we're going to talk about the red blood cell. Okay, so that's our objective today. So what we're going to do is we're going to talk about the structure. We're going to give you a general overview of its lifespan, hemoglobin, and blood types. Okay, so the structure of a red blood cell. Okay, the first thing you should know about a red blood cell, it is a biconcave disc. Okay, so basically I cut this red blood cell in half. Okay, so I've cut it in half. Okay, the width of the red blood cell is about 7.0 to 8.0 micrometers. That's micrometers. Okay, so that's tiny. Okay, so general impressions. We can say it is small. 7 to 8 micrometers. Okay, and it is biconcave. Okay, so basically biconcave, it bows in on both sides. So if you look at a red blood cell on that side, kind of looks like that. So it's really skinny in the middle and a little bit thicker at the ends. Okay, that's what we got, biconcave. And I think the most important of this, it is anucleate. So there is no nuclei. N. I was just going to write N as N. In a red blood cell. Now, this is really important because you know it's a mature red blood cell when you see that they have no nuclei. Now, if you were to ask me, are there red blood cells that do have nuclei? And I will tell you, yes, there are red blood cells that have nuclei. They're immature. Okay, they're called reticulocytes. When you see reticulocytes inside of the blood, and you will see the occasional reticulocytes, but when you have this state called reticulocytosis, that means you're producing blood cells too fast, and there's something going on. Okay, but this isn't a disease class. We're not going to go into that. Okay, so that's the general structure. Okay, the interesting thing about the red blood cell, it has a high surface to volume ratio. Okay, so that means there's more surface. There's more surface than there is volume. Okay, and what this is, it allows for quick absorption, and release of oxygen / carbon dioxide. Okay, we're also gonna find out later is that red blood cells, hemoglobin, which is inside the red blood cells, hate oxygen, and it's a pain in the butt to load hemoglobin with oxygen, and as soon as it can, it kicks it off as quickly as possible. So that's another thing. Because of their structure, red blood cells are incredibly stackable. Like coins, and they are very flexible. Okay, so the 7 micrometer. Let's do that to where it's actually legible. The 7 micrometer red blood cell can squeeze into a vessel with a 4 micrometer diameter. Yeah. So almost half. It's almost half. A red blood cell, which is about seven micrometers across, can fit into a vessel that is about half its width. So it's very, very flexible. And in fact, the loss of flexibility is one of the signals to the body that a red blood cell is too old. Because if it can't flex, if it's not flexible anymore, the spleen will sequester it and get rid of it. So that's the shape and function of it. Okay. Now the lifespan. The lifespan. The lifespan of a red blood cell. Okay. So we already know where they're born. They're born in the red bone marrow. Okay. That's where they are. Okay. And we already know what stimulates them. Stimulated by erythropoietin. From the kidneys. That's what stimulates them. The ironic thing and the sad thing about them is they have no means of self-repair. Okay. So they have a very short lifespan. Okay. And when it is time for them to die, they will be destroyed in the spleen. And how long is this? Approximately 120 days. Okay, about 120 days. That's how long the average blood cell will live. Now you might ask, what if you don't have a spleen? Well, then the liver does the job. Okay, so that's it. The lifespan, 120 days. Where are they born? Bone marrow. Where do they die? The spleen. All right. So the inside, the actual working part of the red blood cell, is called hemoglobin. Okay. In med term, hemo means blood. You know, globin means protein. Okay. So hemoglobin literally is the blood protein. It is the main intercellular component of the red blood cell. Yeah. Right. Hemoglobin itself is a quaternary protein. It is a quaternary Okay. So what this means is you have three foldings. You have three stages of a protein. Remember, stage one. So stage one is the chain of amino acids. Number two is the twisting of the chain. Third is further twisting. Into globs. Okay, makes it globular. And then quaternary, which is right here, this is two or more proteins hooking up, basically, intertwining for final shape. Okay, so those are the four. Okay. So when we say we got a quaternary protein of hemoglobin, okay, there are 4 protein subunits in hemoglobin. There are 4 protein subunits. Okay, 2 alphas and 2 betas. All right. Each one of these 2 alphas and 2 betas has a molecule of heme in it. Okay, each has a molecule of heme. Okay, this is important because this will turn into bilirubin. And bilirubin will eventually make bile. And if it gets out of control, it can cause jaundice. if it gets out of control. The important part of this is this heme. It contains, okay, 1 iron atom. Okay. So we have 2 alphas, we have 2 betas. Okay. Each one of those strands, so each alpha strand and each beta strand has a heme, has a molecule of heme. Attached to every one of those hemes, there is an iron atom. Okay. So in every hemoglobin molecule, there are 4 irons. The iron can bind to 1 oxygen. So every hemoglobin molecule can carry 4 oxygen molecules. Okay. It is this iron allows the binding of oxygen. Okay. And carbon dioxide. That is the iron. Okay. Now, little quiz. Why is your blood red? Why is oxygenated blood red? Deoxygenated blood, or quote unquote venous blood, quote unquote deoxygenated blood, dark red. And it is this. Okay. And you've all seen what happens when you put oxygen with iron. You get rust. Okay. When you oxygenate the hemoglobin, you're basically rusting it. You're oxidizing it. Rust is just the oxidization of iron. Okay. So when you add oxygen to iron, it rusts. It turns a bright reddish orange color. And that's what gives oxygenated blood its bright red color. It's the way the light reflects off of the oxygenated iron. That's it. Probably didn't want to know that, but I think it's cool. I really think it's cool. All right. So basically what we have, so we can just draw this out really, really quickly. Okay. So we have an iron, we have an iron, we have an iron, we have an iron. Okay. Okay. So around this, attached to this iron, okay. Okay. This is our heme in the red. Okay. And then around that, you've got 2 alpha chains and you got 2 beta chains. So we're going to do blue. We got this weird, let's go call this the beta chain. You might have another beta chain. Beta. And then you'll have alpha chain. Might do some weird stuff. That's our alpha chain. And then here you have the alpha chain. That's pretty much what you got. Pretty much what they got. They all connect to each other somehow. Okay, so they're all held together. But basically that's what we have. We have four chains, 2 alphas, 2 betas, 4 hemes, and 4 irons. I think the 4 irons and the hemes is the most important part of it. I don't think it's so important that you understand that we got 2 alphas and 2 betas, because clinically that's not significant for you guys. Okay. All right. So we're going to go over again. We're going to go over ABO blood groups. The ABO blood groups. Now, there are more blood types than the ABO groups. Okay. But the ABO groups are the most common. By far the most common. Okay. So from this ABO, we can get basically 4 blood types. Okay. You have type A. You can have type B. You can have AB, and you can have type O. Okay. So now, what determines what blood type you have? What type you have? And so basically, if we have a red blood cell right here, we have a red blood cell. Okay. What it is on this red blood cell, there is a marker. There is a marker. I'm not going to draw it like that. I'm going to draw it like a little here. It's got this. And let's say it has an A on it. Okay. And so what this A is, this is called a surface antigen. Okay. It belongs on your cell. It belongs on your cell. Okay. Now your body, we have, there are predominantly 3 surface antigens, really. There's A, there's B, and then we're going to talk about this one later called the RH antigen. So if you only have an A surface antigen attached to your cell, this means you have type A blood. Okay. And so you're like, well, what determines this? Let's say mommy and daddy, not mommy. So mommy and daddy, they really love each other and they decided to have a baby. And let's say mommy was type A blood and daddy was type A blood. Okay. You're only going to get one of these markers. Okay. So if mommy is AA, daddy is AA, you're only going to get one here and you're going to get one from daddy. So this means you only have type A surface antigen. Okay. That's where your blood comes from. That's how it comes. Okay. So now let's say that you have your red blood cell, okay, and on it, it's B, okay, and this is your surface antigen. Okay, this is type B blood. Okay, so how did this happen? So we got mommy and daddy. Okay, so this could be a little bit more complex. Okay, so we can do scenario 1 where mommy is BB blood and daddy has got nothing but B's. So you get one here and you get one here. Okay, so that's it. Mommy and daddy, that you're going to be type B. Okay, that's scenario 1. Scenario 2 is, let's say mommy plus daddy. And I'm trying to be sexist. We're talking to this sexualist. This is, biology. So let's say mommy is AB and daddy is AB. Okay. So that means you get this B and you get this B. It comes down, you are type B. And we get scenario 3, mommy plus daddy. Okay. Let's say mommy is A, B, and daddy's type O. That means you only get this one from mommy. So this is type B. Okay. So that's basically the third scenario. Okay, it's a little bit more complex than that, but you get the gist. Okay, so now, we've already done A, we did B, so let's say we have a cell here. Okay, you got a surface antigen here that's A, you got a surface antigen here, so B. Oh, look at this, we got 2 different surface antigens. Okay, A and B. This means you have type AB blood. Okay, type AB. Okay, so here, so how this happens, mommy and daddy. So mommy can be A. Let's say mommy's AA and daddy's BB. Okay, so you're gonna get an A from mom and a B from dad. That's gonna give you type AB. Okay, or the second scenario, you have mommy and dad. Let's say she's AB and dad is AB. That means you get A from dad, A from mom, and B from dad. This is going to be AB. Or you can get B from mom and A from dad. That's it. Okay. Now, mommy can also be BB and daddy can also be AA. So it doesn't matter. You just see how it combines together. Now, here's the thing. So let's just say here, I have mommy and daddy. Let's say she's AB and dad is O again. Baby can never be AB. Baby can't be AB. Baby can only be A or B because daddy's got nothing to give. Nothing to give there at all. Okay, so keep that in mind. All right, so then the last one. What? No surface antigens equals type O. Type O. Type O. It's a type O. Okay, yeah, kind of mistake. So this means mom has to be O, dad has to be O. All right, you have to have no surface antigens. Okay, so that's type O. So let's take this a step further. Okay, let's take this a step further. Let's say we have here surface antigen A. Now in the blood, also in the blood, you have these things called anti-B. So if you have surface antigen A attached to your bloodstream, okay, that means inside of your bloodstream you have something that will attack the opposite blood type. Okay, so if I give somebody B blood, if I give somebody type A blood, and if I have somebody who has type A blood and I give them type B blood, they're going to attack it. Okay, it's called transfusion reaction. It's very bad. Okay, if I have a blood that's got surface antigen type B, that means it's going to have anti-A. So again, B, the opposite of the B antigen, is A. So if I have that, if I have somebody who has got B blood and I give them A blood, they're going to attack it because in their blood there is something that will attack A blood. Now, if I have somebody who has AB blood, what? No anti-antigens. Lucky suckers. All right. You can give, oh, I said type A, type A. That should be type A, type B. Okay. These guys got it made in the shade. Okay. So they have no anti-antigens. Okay. So you can give these guys or gals, type A blood. You can give them type AB blood, and you can give them type B blood. Okay. Perfect. Great. Absolutely great. So these are universal receivers. Okay. Now, let's look at the guys, let's look at our subjects who get screwed over the most. Our type O's. But they have anti-A, and they have anti-B. Okay, which means they ain't getting nothing. They can only get type O blood. Okay, now type O bloods, when you clean it, got no surface antigens. They can give their blood to anybody. Type O's are universal donors. Okay, universal donors. Perfect. Absolutely perfect. Okay, ABs, universal receivers. I before E. Okay, receivers. Okay. So that's what we got. Now we got to throw another monkey wrench into the gears. Okay, the RH blood group, RH antigen. Okay, it's called RH because we found it on rhesus monkeys first. And basically, what this is the plus or the minus. You've seen that. So if you have somebody who has surface antigens A and surface antigen RH, that means they are A positive. They are A positive. You have to pay attention to this. Because that means if you have somebody who is, okay, let's explain it like this. If you have somebody who is just A, okay, and you give them a positive, their body is going to attack this red blood cell. It's going to attack it because it's got that RH antigen on it. So if somebody, this is recipient, and this is donor, okay, that RH, the presence of that RH means that the white blood cell of the recipient is going to attack that. It's going to attack that red blood cell. All right, so you have to pay attention to that. So if you want to lay it down, okay, so you have A, okay, A and A positive. We have B and we have B positive. That's me. You have AB, AB positive. You have O and O positive. A lot of people will say negatives. I don't like that convention, because if it's not there, it's not there. You don't need to say that. But if the positive means it's there, if it's not positive, it's just not there. Okay, so your type B means you don't have it. Okay, so now the thing about this is, you have to look at this. You still want to look at here, this one. This is still the universal donor. Okay, the universal donor, because there's no RH. They can give their blood to anybody whatsoever. There are no surface antigens on this whatsoever. This right here would probably be the worst. O positive would probably be the worst blood type to have ever. Okay, then we have AB positive. This is definitely the universal receiver. Okay, because you might like, well, what happened if you give them AB negative blood? It doesn't matter, because there's no antigen there. It only becomes a problem if you try to give somebody who has no RH an RH positive. Okay, you can't do that. You do not go this way. You cannot donate this way. Do not donate backwards. You can donate forwards. Okay, well, let me do that in different color. Actually, that makes a lot of sense. That's actually really cool. I just thought about that. Okay, let me do it in red, because red means stop. Okay, no, you do not donate in this direction. Don't donate in this direction. Okay, green means go. You can donate in this direction when you're talking about RHs. Okay, you can donate from quote unquote negative to positive, but you don't want to donate from positive to negative. Okay, and that's how that cookie crumbles. All right, so the last thing we're going to talk about, actually no, we don't. I think we're done with this. We already talked about that. Sensitizing. Okay, good. So those are the blood groups. Now, the thing about blood groups is you're just going to have to work it out. It's one of those things that's, it's a little bit of mental gymnastics, because it's not how we really like to think about things. Okay, so just go through it a couple times and you'll figure it out. So just remember that if you're type A, you can only get type A blood. If you're type B, you can only get type B blood. If you're AB, you can get A and B and AB blood. If you're O blood, you ain't getting jack. Okay, you just got to get other O bloods. Okay, so that's, once you tackle that one, that's cool. Then you got to say positives. Positives can take from negatives. So negatives can give to positives, but positives can't give to negatives. I think that's the best way to say it. Okay, before I confuse you anymore, I'm going to cut it off right there.